Good afternoon, everyone. I'm Subash, and firstly, I'd like to thank the organizing committee and Corteva for giving me this opportunity to present. And after all delays and cancellation, I was able to make over here last night at 1 a.m. So now moving into my presentation. Okay. So the title of my presentation is a high-throughput phenotyping improves the predictive accuracy for FHB-related traits in the winter wheat. So I'm currently working with Dr. Sunish Sehgal, a winter wheat breeder at Winter Wheat Innovation Lab. So moving into my presentation, a slight background about what, as you all know, that wheat is one of the most important crops that is grown throughout the world, and they are consumed in different forms. And so I'll be talking about one of the major diseases that wheat is being infected throughout the world, and that's a fungal disease caused by Fusarium gramineum, also known as Fusarium Head Blight, and we can also call it as a scab. So, So here, over here you can see a healthy spike, what it is supposed to look like, and over here you can see an infected spike of the FHB. And basically, what it does is it infects the spikelet, and it leads to the chlorosis. And you can see, over here, you can see the green spikelet, and down here as well. But over here, you can see the spikelet seems to be mature, but it's not mature. Basically, it's the symptoms that fungus is showing, and it also produces toxins known as DON, which is known as deoxynivalenol, and causes a lot of losses in terms of money, since it's a toxin, and there is a severe impact in the health for both animal and human feeds. So USDA has also ranked this disease as one of the most devastating crop disease. So once these symptoms is observed and you trace this head, you will be getting a mixed seed, which is, over here you can see the infected seed, and these are the healthy seeds. So basically what we call them as, they are known as Fusarium damaged kernel, FDK, and this is basically a healthy seed, what it's supposed to look like. So this is the problem, and developing a FHB-resistant line is one of the major goal, and one of the, I think, most effective way to minimize the losses. So taking into that consideration, so we do have a scab screening nursery in our breeding program every year, and it comprises of different materials we screen it every year. So we have the advanced breeding line from SDSU winter wheat breeding program, that's CPT, AYT, Elite, and PYT, comprising of almost 400 lines. Apart from that, we also have the NRPNs and SRPNs, that's Northern Regional Performance Nursery and Southern one, and we do screen some Durham lines and lines from private companies as well. So it's make altogether almost around 1,100 lines, and we do it in 3 to 4 applications, making almost around 3,000 to 3,500 lines in every year. So still a progress in FHB resistance has been limited. So one of the major challenges for progress in this disease is the phenotyping of post-harvest FHB traits in short time. So if you know about winter wheat breeding program, if anybody have worked over here, so I would like to show you a cycle how it really works in our program. So talking about the inoculation, we go for inoculation in the month of June. And after 21 days of inoculation, we take the data for that disease, that's what we did in the field, that's visual disease index. And after 3 weeks of that, the plants are ready to harvest, and we go for harvesting. And after 3 to 4 weeks, we have to go for replanting. So basically, breeders have just 3 to 4 week time period to make all the selections, like which lines are good and which lines are bad. But meanwhile, he has only the one traits in his hand to make the selection, that's visual disease index. But there are 2 other important traits that need to be taken into consideration while making the selection. That is FDK and DON content in the samples. Basically, it's practically not possible to do that within that time period. So breeders are making selection based on only visual disease index. So that way they are not having the accurate selection, and progress has been limited. So apart from time, there is a biasness of FDK rating. So basically, as of now, what we are doing, we are just simply seeing the samples, and based on our experience, we are trying to say, oh, it's 10%, it's 20%, it's 30% FDK in there. But if I rate, I will say it's 30%. If other person rate, it's going to be 40%. So it's kind of biased, and we don't have the accurate phenotypic data for FDK. And for DON, we generally send our sample to NDSU for estimation, and they almost took it around, give us data back after 4 months. So there is no way you can use this data. So my major goal was can we make this faster, accurate, and cheaper? and can genomics and phenomics be a good tool to overcome this problem? So taking that into consideration, I carried out an experiment in major 4 steps. The first step was planting inoculation and visual waiting. Over here, we go for inoculation when the plants were at 50% flowering stage. We took the optimum moisture was provided, and we took the data at 21 days after inoculation, and those lines were harvested for FDK. Then after that, we use a QSorter for FDK estimations. So I'll be explaining more about QSorter in the other slides. So basically, I'd like to thank Dr. Gill for his mentorship, for teaching me how it works, since I was joining this lab and he was about to transition from Ph.D. to postdoc, since he's doing his postdoc in University of Minnesota now. So basically, this is an AI machine. How it really works is that I will show you a blueprint over here. So over here, you have a feeding hopper. So you put your FDK infected sample over here, and it has a build. So the build basically transports single kernel at a time, and there are 2 different cameras, that is NIR and RGB cameras. And single kernel are taken through this build, and the camera captures the picture every time the kernel passes through that area. So basically, this works at a speed of 30 to 40 seeds per second. And these data are collected and further analyzed over here using different machine learning models. And at a real time, the samples are sorted into 3 different bins. So the first bin is for the unclassified seeds. Sometimes you may have dirt, soils, or weed seed in there, and the broken seeds. So those seeds will go over here in the first bin. The second bin is for the infected seeds. That's the FDK for our case. So all the FHB infected seeds will be going into bin 2. And the third bin is for the healthy seeds. So once this sample is run, and we'll be getting 2 FDK data, because since we're having 2 different cameras, that is NIR and RGB cameras, so finally we'll be having 2 different FDK data for our same sample. Then moving to the step 3rd, I also tried to use the hyperspectral camera. I basically used the proximal handheld hyperspectral camera and try to take a picture of the infected seed and try to see that if I could predict or estimate DON content in the sample itself based on the seed image of that hyperspectral camera. And in the 4th step, I used different genomic selection models and phenomic selection to try to see and predict and validate. So moving into my results section, first I used the single thread genomic selection model. So over here, I use 5 different models and try to predict the threads, since I told that we have 2 different FDK rating from QSorter, that is from VQV, that's a RGB camera-based FDK, and NIR is NIR camera-based FDK. And FDK-V refers to the FDK data that is often from the human rater. So, and DON basically is that toxins. So once we run this model, and what we observed that we are having the highest prediction accuracy for the phenotypic data of FDK that was obtained from QSorter itself. So we are getting the accuracy of 0.11 for the FDK visual, while in case when we use the FDK from QNIR, we are getting the accuracy of 0.39, which shows a significant improvement in the prediction accuracy. And moving on to the multi-thread genomic selection models, we also collected the other secondary traits like plant height and heading dates, and try to incorporate into different combination and try to predict the DON content in the samples. And we also use the phenomic prediction model simply alone, so try to see that what phenomic can do in there. And what we observed that the combination with the FDK data obtained from QSorter was having the highest prediction accuracy of 0.49%. So we took here single thread zblob as a standard baseline, that's 0.31%. And apart from that, we also try to see, like, we already have the hyperspectral information from all the samples. So what we did, we extracted the hyperspectral indices and tried to incorporate that in the genomic prediction model. And we selected the top 10 bands, which were showing highest correlation with our trait, that's DON content. And also we tried to use different combination, and we found that a combination of 6 different web bands was having the highest prediction accuracy of 0.43% compared to other, and this is the first time I think that has been done in FHB itself. And over here we did the phenomic prediction using 1D CNN model, and we got the prediction accuracy of 0.45, which is kind of comparable to that of multi-thread genomic prediction model, and it shows that it also has some potential that we can go for some phenomic prediction model to estimate DON content in the sample. And moving into the outcome applications, so basically our results demonstrate that integrating the high-throughput data into our genomic and phenomic prediction models, we can significantly improve the prediction accuracy for these traits, that is FDK and DON. And phenomic prediction model alone integrating hyperspectral imaging has the potential to estimate the dawn as well. I would say like 0.45. It's not that good, still you can do that. And talking about the application part, breeder can use this information and use these models and try to go for the negative selection since the accuracy is not that high. So it's always beneficial to go for the negative selections. So that will be the application part. And talking about the future direction, what I'm trying to focusing now is that we are planning for forward prediction of these traits. And we also have taken the UAV data for our FHB nursery and trying to look how we can predict the disease severity in the field itself by using UAV data. And apart from that, this is currently I'm working on this project using the computer vision model, that yellow V11, and try to annotate the infected and healthy spikelet over here. I think it's not that visible though, but we're trying to train this model, and we're simply trying to take a RGB image using our camera phone and trying to see that if that model can identify the infected spikelet and the healthy spikelets. So that way you can have the accurate disease severity data in the field itself. So you don't have to sit there and count how many spikelets are infected and how many are healthy and do that. So instead of that, we can have just take a picture and get this one. And over here, I try to show that FDK, we do the visual rating. So we don't really count like how many are infected and how many are healthy. So if we could take the picture and the model can itself identify how many are infected and how many are healthy, then we can simply get the FTK percent in that particular sample. So it can reduce and improve the phenotypic data, and we can use that data into our genomic prediction models, and obviously it's going to improve our prediction accuracy. And lastly, I'd like to thank all of my teammates, my advisor, Dr. Sunish Sehgal, and all the collaboration from Geography Department and Department of Plant Pathology and USDA eras, Dr. Bai, Amy, and Paul, and my funding sources, SCAB Initiative, South Dakota Wheat Commission, and my lab itself, Integrated Innovation Lab. And thank you.
We have a minute for questions. Any question?
Great presentation, thank you.
You.
Have you looked at using like grain scan, where you use that software in conjunction with just a normal desktop scanner?
This slide, this
yeah.
one? It was like, I just use my mobile phone and take a picture, and we just feed this into model. So basically we are training, like first we initially annotate by ourselves, like this is the infected, this is the healthy one, and train the model. And once the model is trained, and you just feed the picture in that model, and it will annotate itself like the infected and healthy.
Great presentation. So I have a question about the ground truthing of infected versus non-infected. So maybe I have missed it, but you have this visual evaluation versus the image-based evaluation. But what was the method that you used for ground truthing?
So the ground truth was, for this one, we are doing ourselves for this particular thing. So you told about that ground truth for that FTK visual for QSorter? QSorter?
Yeah, I was thinking about the sorter.
Sorter basically, the ground.
Yeah.
Over here?
Yes.
So the visual was just a rater go and rate their samples.
Yeah, I understand, but you have to have a ground truth. Essentially, you have to be able to tell this was infected versus not, right? So what did you use for that? Okay, so basically this QSorter we trained and we calibrate and try to use the samples that we already knew that there is 10%, there is 20%, and try to see the accuracy of that one. It was like 96, 97%. So that was the ground truth for us. Okay, so you had that mixed sample where you knew the percentage.
Yeah,
okay,
we knew that. How we count and we just add and mixed up and just did that.
Okay, thank you.
Yep.
Let's appreciate the speaker for nice presentation. Thank you.
Thanks.
Subash, Thapa.